Good morning, everybody. My name is Vaishnavi Srinivasaraghavan, and I'm going to be talking about mammary cancer cell manipulation with our newly developed embedded passivated-electrode insulator-based dielectrophoresis. I collaborated on this project with Tyler Sheikh and Phillip Zellner, and under the direction of my advisor, Dr. Masoud Agah. So at VT-MEMS lab, we mainly focus on using MEMS technology to develop sensors for chemical and biological analysis. This particular research that I'm presenting here falls under the bioelectronics subcategory of the bio-MEMS thrust in our lab. DEP is a well-known technique for manipulating uncharged particles using non-uniform electric fields. The DEP force on the particle is given by the first equation here, where which is directly proportional to the cube of the radius of the particle, the permittivity of the medium, the real part of the Clausius-Masoud factor, and the square of the gradient of the electric field. The Clausius-Masoud factor is given by the second equation here, where epsilon p and epsilon m are the complex permittivities of the particle and the medium respectively. Since the permittivity of the particle and the medium are change with the frequency, the Clausius-Masoud factor can change from a positive value to a negative value over the frequency spectrum. This results in either a positive DEP force or a negative DEP force that the particle experiences, and can either move one direction or the other. For the DEP force to be successful in manipulating the particle, it has to overcome this drag force on the particle, which is given by the last equation here. So using these DEP forces, researchers have developed microfabricated sensors to manipulate different kinds of particles, which include beads, bacteria, viruses, and also mammalian cells. The non-uniform electric fields that are a requirement for dielectrophoresis can be created by one of two major techniques. Either Idep, which is electrode-based, or Idep, which is insulator-based. In Idep, electrodes are used to create the non-uniform electric fields, and as you can see here, they are two-dimensional, and as you move away from the electrode in this direction, the strength of the electric field decreases. So the maximum trapping actually happens on the electrode, which is right there. In insulator-based dielectrophoresis, we use insulating structures in the channel, which can be designed in any way to create these non-uniform electric fields. One of the disadvantages of Idep is that there is a lot of heat buildup at these insulating structures due to the large electric fields that are required to create these non-uniform electric fields in the channel. So at VT-MEMS lab, our initial focus was to develop silicon-based Idep devices for particle trapping. Here are some images from our journal papers. We have developed both two-dimensional and three-dimensional insulating structures in the channel to trap particles. And here you can see trapping of beads in the channel in our silicon-based Idep devices. We also developed a process flow for the fabrication of the three-dimensional structures that are easy to fabricate in silicon, but very hard to fabricate in PDMS, using a glass reflow process, which was published in JMEMS. As you can see, instead of having a silicon master, which is usually used for PDMS fabrication, we create a glass master from the silicon substrate, which was initially fabricated using deep reactive ion etching. And using our PDMS devices that were fabricated using this method, we showed trapping of beads, as you can see here. And more recently, we introduced this technique called EPiDEP, which is off-chip passivated electrode insulator-based dielectrophoresis. Here we have the electrodes, which are either on glass or a printed circuit board. which are passively coupled with the Idep device, which is right here. So the device can be fabricated separately and then coupled with the electrodes by using just paper clips. And since this is not directly bonded to these electrodes, they are reusable. The electrodes are reusable, and you can make new Idep devices to go on top of the same electrodes. As you can see, we use this technique to trap different strains of bacteria here. Here you can see E. coli being trapped on the insulating structures. We also showed the trapping of Staph aureus and also showed separation between bacteria and beads. One of the disadvantages of this device is that because you're capacitively coupling through the glass cover slip here into the Idep device, the electric field strengths in the channel reduce greatly. So, as this is a simulation that shows the electric fields in the channel as a function of the thickness of the passivation layer. And as you can see, a thinner passivation layer greatly enhances the electric field strength in the channel. So, this is our new EPiDEP device, where our electrodes are embedded in the channel but separated from the insulating structures by a very thin passivation layer of PDMS, which is spun coated on the electrodes, and this is 5 microns thick. The insulating structures consist of a vertical column of microposts, which are 100 microns in diameter and separated by 50 microns between each other. And the electrodes are 400 microns in width and separated by 600 microns, which are very close to the insulating structures to improve the electric field strength right at the insulating structure. So, Tyler recently published a paper in Analytical and Bioanalytical Chemistry demonstrating the use of these devices to trap different strains of bacteria and also separate live and dead E. coli. Here, we show the fabrication of these devices is quite simple, and we first fabricate the electrodes on a glass substrate by evaporating them onto the glass substrate. Then we spin a thin layer of PDMS onto the electrodes, which is 5 microns thick, and then bond the glass layer to the PDMS channel layer by plasma bonding. The PDMS channel itself can be fabricated either from a silicon master or a glass master using either of our fabrication processes. The cells that we used in this study were the MDA MB468 cells, which are a metastatic breast cancer cell line. These were purchased from ATCC and grown in T75 flasks. So we harvested them from confluent cell cultures and centrifuged it at 1000 RPM and resuspended them in a DEP buffer, which consists of sucrose and a little bit of RPMI. The conductivity of this DEP buffer was 210 microsiemens per centimeter when we measured it using a handheld conductivity meter. The cells were also stained with calcine, which is a green fluorescent dye, to enable fluorescence microscopy during experiments. This is our experimental setup. So we used a syringe pump to induce flow of the cells through the channel. We set the flow rate at 50 microliters per hour, and we excited the electrodes with a 300 volt peak to peak signal, and we did a frequency sweep from 100 kilohertz to 3 megahertz to investigate the effect of cell frequency on cell trapping. These images, okay, I had a movie to show you the trapping of the cells. Okay, so this shows before the cells start to trap, you see them freely flowing in the channel. when the voltage is switched on, the cells start to trap onto the insulating structures, and we let the voltage stay on for 30 seconds, during which continuous trapping happens. And once we turn off the signal, the cells are released from the structure. So these images show trapping of cells at 3 different frequencies, and as you can see, there is good trapping across the frequency spectrum. To quantify the trapping at different frequencies, we counted the incoming cells and the outgoing cells in each movie, and calculated the capture efficiency as the incoming cells minus the outgoing cells divided by the incoming cells. And as you can see here, this plot shows that there is good trapping over a large frequency range, which is over 90%, and the maximum trapping was observed at 500 kilohertz and was 97%. So in conclusion, we demonstrate trapping of MDA MB468 cells on our EPiDEP devices with very high efficiency. This inspires future work in characterization of progression models of breast cancer, as well as selective trapping of cancer cells in these devices. So we thank our funding source and you for your attention.